my name is Siti Farahani binti Alafiz. My matrix number is 202499. So today I'm going to demonstrate a step-by-step technique streaking for single isolated colony. So let's move on to our experiment. Plate technique is technique used for isolations of pure culture from a mixed populations where the number of organisms will be reduced in order to get a single isolated colony. So in this experiment, I'll be using one part of Serratia marcescens and 3 parts of Morgellopus luteus, and we'll be isolate these 2 bacteria into a single colony. These are materials that I will be using in this experiment. First, we have nutrient broth culture. Second one, we have agar slant. And the third one, we have a nutrient agar plate culture. In this experiment, I'll be using a quadrant streak method where you can see I have divided the plate into 4 equal parts and labeled each quadrant 1 until 4 at the bottom of the plate. Next one, I have my inoculating loop and also candle as substitute to Bunsen burner. So let's move on to next part. First of all, make sure to sterilize the inoculating loop by flaming it to candle flame for a few times and let it cool for 10 seconds. The next step is uncap the tube and flame the neck of the tube by passing them through the flame once while still holding the inoculating loop. Don't put it down, like what I'm doing right now. Next is obtain a loopful mixture bacteria from nutrient broth culture and reflame the neck of the tube to sterilize it. With the loop containing mixture of bacteria, wrap a small circle into the agar plate on quadrant 1. Close the lid of agar plate, reflame the loop and let it cool again. We have to make sure to cool down the inoculating loop before touching the agar plate because we don't want our bacteria culture dead. Next, from the quadrant 1, streak the agar across the quadrant 1 to quadrant 2 to 5 to 6 streak. Reflame the loop again and let it cool for a while. Make sure to streak only within your own quadrant because we want to reduce the number of organisms from the previous streak. Moving to the next, from quadrant 2, streak another 4 to 5 lines to quadrant 3. Reflame the loop and let it cool. From previous streak, streak another 4 to 5 lines across quadrant 3 to quadrant 4. Lastly, I'm going to incubate the agar plates containing mixture of bacteria for 48 to 72 hours at 25 degrees Celsius in an inverted position because we don't want the water from the condensation process of the agar may fall on our bacteria culture. So after I have incubated the culture, we can see a well-separated colonies had developed on the surface of nutrient agar plate. The red pigmentation represents the S. marcescens and the yellow pigmentation represents the M. luteus. Next step is, I'm going to transfer each of these bacteria into agar slant to isolate it. The first step is, we have to sterilize the needle by flaming it and let it cool for 10 seconds. Obtain a single colony from agar plate. and transfer it to agar slant that has been labeled beforehand for each of the bacteria. Place at the base of the agar slant and withdraw the noodle in a zigzag motion. Next, flame the neck of tube and reflame the needle. And lastly, I'm gonna incubate all the agar slants at 37 degrees Celsius for 18 to 24 hours. These are a few of the expected results that we can get from the experiment.